Good afternoon, Mr. Chairman and dear colleagues. So today I will present you two pro-apoptotic proteins, BNIP3 and NIX, in pancreatic ductal adenocarcinoma and their regulators. I will start with the main focus of our project, which are three important aspects of pancreatic ductal adenocarcinoma that I will refer as PDAC from now on. First, apoptosis, which is a programmed cell death. Apoptosis normally maintains tissue homeostasis. However, evading apoptosis is one of the hallmarks of cancer. In the case of PDAC, it is highly resistant to apoptosis. Then hypoxia, which is a limited oxygen condition. PDAC is known as one of the most hypoxic tumors. When oxygen tension is around 52 millimeter mercury in healthy pancreas, here, in PDAC, it's actually less than 3%, 3 millimeter mercury. So, and lastly, it's p53, which is a well-known tumor suppressor, also known as the guardian of the genome. And it is important for us because 60% of the cases, it is mutated in PDAC. Let me give you a brief outline about how these three concepts are related. Normally, under normal conditions, cells can tolerate hypoxia up to a point, but when it persists, cells go to apoptosis and die. Hypoxia also stabilizes p53, so it can transcribe genes that regulate apoptosis. So there are many pro-apoptotic and anti-apoptotic proteins that plays a role in apoptosis to maintain cellular homeostasis, but not all of them are affected by hypoxia. So BNIP3 and NIX, also known as BNIP3-like protein, can be induced by hypoxia and p53 as well. In the end, apoptosis and p53 together suppress tumor formation. So, these are selected articles that shapes our hypothesis. So in this article, they found that BNIP3 is silenced in PDAC due to hypermethylation, which is an epigenetic phenomenon that reduces the expression of a gene. Meanwhile, our group published the loss of BNIP3 gives PDAC a more aggressive phenotype, worsened prognosis, and chemoresistance. And this article is also about NIX, showing that the connection between p53 and NIX in hypoxia, also NIX's absence promotes tumor growth. And the last one is showing that p53 also can regulate and suppresses BNIP3 and protect against hypoxia-induced apoptosis. So in the light of the literature, we have some questions. We know that BNIP3 is lost, therefore apoptosis is inhibited, and in the end, PDAC is become more aggressive. Well, we still don't know what NIX does. So whether they work in an orchestrated manner or opposite, we don't know that. So therefore, this was our first aim to find out. And the second one, we know that NIX is regulated by p53. But what happens when p53 is mutated? As I mentioned earlier, is 60% of the time it is mutated in PDAC. In order to find out, we performed a series of experiments to see gene and protein expression of BNIP3 and NIX, also the methylation pattern of NIX. We also evaluated the effect of hypoxia and cell viability. As the answer for our first question, we observed that NIX is overexpressed in chronic pancreatitis and PDAC tissues compared to normal pancreas, whereas BNIP3 expression is actually reduced when we compare to normal pancreas. So when we correlate these two proteins' gene expressions, we actually saw that there is an inverse relationship between them. So when NIX is upregulated, BNIP3 is downregulated. Here we wanted to see protein expression of NIX in tissues. Compared to normal pancreas, NIX is actually overexpressed in chronic pancreatitis and pancreatic cancer tissues. Even though in pancreatic cancer tissues, the stromal component is quite dense, but existing cells still express NIX. Then we investigated whether the expression of NIX is also epigenetically regulated like BNIP3 is. However, we couldn't find any significant methylation differences between normal pancreas and PDAC. So the expression could be independent from the epigenetic regulators. In this one, we also wondered what happens during the precursor lesions of PDAC. And when we saw that, we actually saw that in late precursor lesions, NIX is expressed more. So I know this might look quite overwhelming, but bear with me. So we wanted to see BNIP3 and NIX expression under hypoxic conditions. Since we can't control the oxygen level in tissue, we went with an in vitro approach and used 8 different PDAC cell lines and grew them in normal and hypoxic conditions. Then we checked protein and gene expression levels, protein and gene, and saw that NIX expression increases with hypoxia. Although there is an increase in most of the cell lines, ACPC1 and CAPAN1 cell lines, this increase was more significant. So we wanted to see if NIX expression affects the chemoresistance. We selected these 2 cell lines. We silenced NIX, treated cells with 2 most commonly used chemotherapeutics, fluorouracil and gemcitabine. However, the absence of NIX didn't affect the chemoresistance significantly. So chemoresistance could be independent from NIX expression. Lastly, I wanted to show you these results, which took our research further. Here you see 2 consecutive slides. One of them is used to detect NIX, and the other one is P53. Just a tiny reminder that when P53 is mutated in tissues, that means that we can actually stain them. So if it's wild type, we can't stain them because it gets degraded. Here you can see strong NIX staining. When we check the P53 in the consecutive slides, we see actually the wild type cells. But when we check the mutant ones over here and compare with the NIX, we can see that the expression was reduced.
Sayın Süer, zamanınız dolmak üzere.
Tamam.
Toparlayabilirsiniz.
So in conclusion, mutant P53 might be incapable of inducing NIX expression. In order to confirm that, we are now doing in vitro experiments. In summary, we have found that expression of BNIP3 and NIX are inversely correlated. NIX expression is higher in PDAC and its precursor lesions. NIX expression is increased under hypoxia, and P53 mutation hampers the NIX expression. In vitro experiments are now in progress to see BNIP3 and NIX's expression and how they related with P53. So I would like to thank my supervisor, Mert Erkan, and my lab members, and then our collaborators to help us to convey this research. And thank you for listening.
Teşekkür ederiz.